A very warm good morning to all of you. Once again, I welcome you all to this short-term training program of NAHP Cast. So now I request my coordinator to please welcome both the dignitaries.
Thank you so much.
So today we are fortunate to have Dr. Archana Sachdev ma'am with us. So and now I request Dr. Anil Dahuja sir, Professor Division of Biochemistry, to please introduce these speakers.
Thank you, Dr. Ranjan, for giving me this pleasant duty of introducing today's speaker, Dr. Archana Sachdev. She is really a respectable figure in the Division of Biochemistry and in the overall all the ICR system, and known for her teaching ability, the research caliber, and she has been recipient of the Best Teacher Award of IRI as well as the ICR. And in Division of Biochemistry, the many courses, they are opted by the students because she is involved in the teaching of those courses. So such is the persona of today's speaker. And not only the teaching, but in research also, she has equally good contributions. She has handled many externally funded projects from NATP, NAIP, NASF, very what we say, and the very rich projects. The funding, it was really remarkable in those, and equally remarkable were the contributions, the research contributions. And she is involved in the area of nutritional quality enhancement. And she, throughout her career, she's working on development of, nutritionally rich soybean crop. So initially, she started with the development of high oleic acid soybean, and then she moved on to development of low phytate soybeans using modern tools of molecular biology, whether it was initially the RNAi, and then she moved on to CRISPR-Cas technology. And she has been successful in developing various constructs for the development of low phytate soybeans. And all these constructs now, they have been filed for patenting. She is the recipient of those patents as well. And apart from this, she is a great human being, and we all respect and love her. Thank you very much. So with these few words, I invite Dr. Archana Sachdev to please deliver the talk.
Thank you so much. It was wonderful. This introduction really made my day. I didn't know that there was. Thank you so much. Good morning to you all. Thank you for this opportunity, and thank you for the wonderful words. Really too exaggerated, but really made my day. So I shall be speaking on this new branch of genomics. You've been dealing with genomics, proteomics, metabolomics, phenomics, everything. But now there are offshoots of these genomics, for example, epigenomics, nutrigenomics. So today's topic is, we are going to concentrate on this topic, nutrigenomics. It's a very potential science, nutritional science, and it has its seeds way back in the time of Lavoisier, that is the 18th century. Look at the vision of Lavoisier, this French nobleman who had said long time back that nutrition has often been the subject of conjectures and ingenious hypotheses, but our actual knowledge is so insufficient that they are only used to satisfy our imagination. If we could have some more facts to supplement them, then we could have applications in medicine. So nutrition and health had found a correlation way back in the 18th century. That was the vision of the man. Now, throughout the 20th century, this nutritional science, it was basically focusing on deficiency diseases. People were trying to find out what are the vitamins, what are the minerals with which we can alleviate these deficiencies. So that was the major focus. And slowly, as industrialization came in, mechanization came in, lifestyle started changing, the whole outlooks of nutrition also started changing. And the developed world shifted to very dangerous diseases, that is overnutrition, obesity, type 2 diabetes, which are highly prevalent in today's time. This became the focus of modern medicine, and the nutritional science now changed to health, development, and growth. So this is the paradigm shift. First from deficiency diseases, then overall growth of human beings. But in the past decade and a half, we have seen a very big leap in the nutritional science. The shift is now from epidemiology and physiology to molecular biology. How? We'll just see. There have been factors which are responsible for this shift. The most important one was the completion of the Human Genome Project in 2003. Now this provided or opened a wealth of new genetic information, and this information we got so much that we are now feeling short of hands to explore this wealth of information. Now, what is the importance of these genes which have been explored by the techniques of genomics? How are they related to nutrition? All these things are to be discovered now. There has been, the other factor is the recognition that micronutrients as well as macronutrients, they are potential dietary signals. They influence the programming in the cell, the metabolic programming in the cells, and they have an important role in controlling the homeostasis of the cell. Also, the researchers have started to recognize that the genetic predisposition of a human being is important in contributing to the main cause of mortality, which is linked to the diet, especially all these diseases like CVD, that is cardiovascular disease, or type 2 diabetes and cancer. Every human being has a different predisposition towards these diseases. If arthritis is there, it's not that it's going to be prevalent after this age in everybody. So everybody is predisposed in a different way to a certain kind of disease, whether it's cardio disease or a bone disease or any other, or related to sugar imbalance. Fourth factor which has been responsible for this shift is the new tools and technologies, the high throughput tools and technologies, which have allowed increasingly detailed molecular studies of nutrition, which was earlier not possible. That's why Lavoisier said that it's all presumption, it's all imagination. If we could get some scientific facts, we could relate how health and nutrition could be linked, and we could bring it to our benefit. At that time, it was not. But with the advent of new tools and technologies, everything is becoming, all that was said earlier, all the vision that those people had in the 18th century started becoming possible and real. And this helped people to change the focus in the field of nutrition. Advances in molecular biology have made it possible for nutrition to undergo a revolution. It's the actual ripe time for us now to make new nutritional strategies, to change the way we think about food, and also to change the way we evaluate the nutritional status and response to those foods that we eat. Initially, we were just eating it for nutritional benefit, to give nutrition to the body. But what are the kind of responses which are elicited, and how are they different in different individuals, that we could not analyze or evaluate. But now we have to make strategies to evaluate that. Now, these tools have made it, they're so precise, they're so accurate that we can even find out subtle changes in gene expression even at single cell level, and we can measure them in a quantitative term and in a real-time term. In a very dynamic way, we can measure what are the changes which are taking place. The entire nutrition-relevant transcriptome can also be studied simultaneously, which was not earlier possible. So such studies have helped to change the focus of the field, and we have now a new field of nutritional genomics or nutrigenomics. And what is nutrigenomics? It is the study and application of nutrient-gene interactions. How does the food that you eat interact with the genes which are present in you? So that interaction nobody had ever focused. We just ate our food and we wanted to see what kind of benefit are we having. Are we feeling healthy or are we feeling sick? But why are we feeling healthy or why are we feeling sick? That interface was not clear. So that interface is what you're going to study through nutritional genomics, and it will be more accurately studied because now we have tools and techniques to study them accurately. Now, how nutrition regulates the pathway of life. We have so many questions to answer. How the diet influences the gene expression. What are the dietary chemicals in our nutrition which affect the health by altering the gene expression pattern? What kind of signature patterns do we observe when we eat a certain bioactive compound? How the interaction of these dietary components with genes take place, and what kind of metabolic profile is produced by those interactions. So you have to see what happens at gene level, at protein level, and then at metabolite level, because there are different levels at which these food materials, when they impact the gene, will produce the results. So we can't just stop at a genomic level or a proteomic level. We have to come down to the metabolite profile level to study the impact of all these dietary components. So overall, it focuses on the net effect of the nutrients on the genome, on the proteome, as well as on the metabolome. The conceptual basis for nutrigenomics can be summarized under 5 basic principles or 5 basic facts, which are: number 1, under certain circumstances and in some individuals, diet can be a serious risk factor for a number of diseases. So 2 things, under certain circumstances and in some individuals, not all, right? Then common dietary chemicals can act on human genome directly or indirectly and alter the expression or the structure of the gene. Another fact which is also clear is that degree to which the diet influences the balance between healthy as well as disease state depends upon what kind of genome or genetic makeup does the individual have. So if you come down, everything is individual based, right? Some diet regulated genes are likely to play a role on the onset or progression or the severity of chronic diseases. So based on this, dietary interventions can be used. Dietary interventions can be used to prevent those diseases or to reduce or cure those diseases. And by dietary interventions, we can come down to a very personalized nutrition level. Because it is individual to individual base, so we come down to a personalized level in which, based upon the individual's predisposition to that disease, you can devise his or her diet, and he or she can ultimately reduce or can feel the reduced effect of those diseases, and we can cure those chronic diseases as well. So nutrigenomics has received a lot of attention in the recent times because of its potential for curing these diseases, including cancer, through small but very highly informative dietary changes. And how are they informative? Because we are going down to an interface which is genomic, proteomic, as well as metabolomic to study those dietary signatures. What is the scope of nutrigenomics? It will give you a global view of the basic physiological mechanism which is influenced by diet. It will give you a dynamic real-time view of your metabolic status at an individual level as well. It will identify new metabolic links that have not been seen before. So if you can get an insight into all these, then it will help in understanding how nutrition influences metabolic pathway and cell homeostasis, how the regulation is disturbed in the early stages itself of the diet-related disease, and to what extent the individual's sensitizing genotype contributes to this disease. Because that person has got predisposition, so he is sensitive to that particular diet. So we can understand at that level that to what extent is that genotype contributing to this disease. So as a result, it will allow very effective dietary intervention strategies. It will help in recovering the normal homeostasis of the cells, and as a result prevent the onset of that disease. So all this information, if we know at the basic level itself, we can prevent or mitigate the onset of that disease in a very effective way through the diet itself, without medicines, through the diet. So if you want to do that, you need to have very clear signatures which will be indicative, yes, this is what is needed for it, and this is how we should proceed. So integration of genomic science with nutrition, and wherever possible with other lifestyle variables, involves finding the right combination of nutrients for each genotype, and this can help in changing the behavior and preventing diseases. It's not that simple as it has been said. There are a number of combinations which change with nutrition and diet, with the lifestyle of the individual, with the life stage of an individual, young, old, whatever, or a child stage, or the nutritional status, whether it is he or she is eating in excess or is suffering from deficiency, nutritional behavior, a preference for something or rejection for something, then a vegetarian or non-vegetarian diet, demographic nutritional changes as per the populations, area-specific deficiencies, could be environmental as well, which particular latitude or longitude are you living in, the household income, whether you have the capacity to spend more or not. All these factors have to be taken because they are all involved some way or the other, in a single way or in combination, to create your impact, to create impacts on your genome. And these studies allow the examination of people at an individual level or at a subpopulation level or at a population level. You have to study in totality because you want to find out those signatures. You want to develop certain markers which will indicate, yes, this is the time to stop or this is the time to start this particular type of diet so that the onset doesn't begin. So it is a complex study. Now, if you consider earlier times, the hunter times or the Paleolithic era, and compare it to the modern times, you will clearly see that initially the energy of the individuals was mainly coming from the hunted food, the raw food that was available. And as the industrialization set in, the abundance set in, the mechanization set in, all the processings took place, availability increased, the taste increased, the lifestyle changed, and there were ways in which we could provide more innovative combinations or types of foods. And as a result, with the change in the lifestyle or slowdown of the kind of activity which individuals started doing, the energy from processed food was more as compared to the raw food. And this is what it resulted in. The calorie intake increased, lifestyle activity basically in lifestyle decreased, life became more easy, input increased, the genes remained the same. Nothing changed. Whatever, the genes are pretty constant throughout. There might be certain SNPs or minor changes that takes place in the gene sequence, but overall, it is one constant factor is the genes, the other things were changing. Nutrition was mainly based on what you find as more tasty, and as a result, because you were so empowered by that taste, that the complexity increased, the processing increased, the innovation in the kind of food that you eat increased. And it resulted in more than 75% of the women turning out to be obese and 58% of the men very close to obesity, with a prediction of 2050 of 23% rise in obesity-related strokes, 34% in obesity-related hypertension, 44% in obesity-related coronary heart disease, and a very alarming prediction of 98% rise in type 2 diabetes. This is extremely alarming for all of us. Now, in order to address this increasing incidence of the diet-related diseases which we are seeing with the changing times, the nutritional research is now investigating nutrition to optimize and maintain at the cellular, tissue and organ levels the cell homeostasis. So, we have to consider biological networks in totality, whether it is at cell level or tissue level or organ level. We have to understand how exactly do these nutrients act at molecular level. Now, it's not just your taste or hunger which you have to satisfy, you have to see what is the impact on the genes which your nutrition is having. And the food that you eat, it can undergo several fates. There can be a pathway A in which it can directly serve as a ligand for transcription factor, and it can therefore impact the gene expression and the normal cell growth. Or it can be metabolized, and the altered concentration of the substrate and intermediates will then impact the gene expression. So, diet, nutrients through metabolism impacting the gene expression and the normal cell growth. Or it may be involved through signal transduction, having an impact on gene expression and the cell homeostasis. So, there are different ways in which the nutrients can impact the cell growth. But what do these nutrients do? They are basically signals. They are dietary signals for the cells. The cell ecology is sensing what you eat, and in the environment of the diet, it will perform accordingly. The nutrients, they are detected by the cellular sensor system that influence the gene and the protein expression, and they subsequently impact the metabolite production. So, you have the changes in these expression. As a result of these changes, the metabolite profile of the individual changes. And these are the signals are what? The nutrients that you eat. And when they are impacting the gene and protein expression, they are creating certain motifs, certain signatures. You have to catch those signatures. The patterns of gene expression, protein expression and metabolite production in response to particular nutrients or nutritional regimes which the individual is taking are called dietary signatures. These are the ones which need to be trapped. These are the ones which are the molecular markers or early signals which need to be trapped. And they are the warning signals, yes, you can go ahead, or no, you should not. This is dangerous. So, nutrients, dietary signals and dietary signatures are the key words for nutrigenomics. Now, this science, it seeks to examine the signatures in specific cells, in tissues and in organisms, and to understand how the nutrition influences the cell homeostasis on the whole. And here the transcription nutrition, the transcriptional factors, they play a very important role because they act as nutrient sensors, the main agents through which the nutrients influence gene expression. We saw that when it entered, what was the fate of those nutrients? It can impact the gene expression through pathway A, pathway B or pathway C. But what is the middle person which is going to take them to that gate where it will alter the gene expression? They are the transcriptional factors. They are the important group of nutrient sensors. And if you see carefully, there are various kind of macro, micro nutrients and other food components. Amongst them, this is the example under those categories. And just see, these are the transcription factors which are dedicated towards response to fat, response to carbohydrates, response to proteins or vitamins or minerals. So, they are extremely important, or they are the sensory system that interpret the information from the nutrients about the dietary environment. And the functional genomic techniques and tools are used at different stages and steps for analyzing those effects or the signatures which are produced. This is your diet. The diet can impact your DNA, or at this stage from DNA to RNA, or from RNA to protein. And these are the gene expression processes which need to capture those changes. And these are the techniques which are used for capturing the changes which are brought about at the DNA level or RNA level or protein level or the metabolite level. So you have the diet, you have the level, and you have the tool to which you detect. So basically you have to detect those signatures. Now which level will you detect the signature? For that level, the techniques are already under play. They are already highly advanced, and you can use those techniques now to very accurately and very sensitively monitor the changes. So different diets are expected to elicit different patterns of gene expression and protein expression and metabolite profile. And nutrigenomics, its job is to identify those patterns which have been referred to as dietary signals. And these dietary signals are examined in the cell or the tissue or the organisms, and in this way the nutrition influences the complete cell homeostasis. This is what we need to investigate, right? Now these functional genomic tools, they can be used in two different and complementary strategies. Either by a traditional hypothesis-driven approach, where specific genes and proteins, the expression of which is influenced by the nutrients, they are identified through the functional genomic tools. These are the tools which are used. And subsequently, you get to know new genes and the understanding of the regulatory pathway through which the diet is influencing the cell homeostasis. And you can use such kind of models to test those changes which are produced as a result of the nutritional intake. And in the background of bioinformatics, large amount of data can be processed. The genomics data or the transcriptomics data or proteomics data, metabolomics. The name itself is suggesting that it will produce a plethora of information that is yielded in the background of bioinformatic tool. We can get a detailed molecular data on interaction between the nutrition and the genome. So it cannot serve in an individual way. It has to serve in conjunction with bioinformatic tools. Otherwise, you cannot process the amount of, you need to process a lot of data to get some kind of a marker, early marker, warning marker, or some kind of dietary signature patterns which can serve as markers. So that needs input of computational studies, which will give you a complete pattern of how this thing is impacting. The second approach is the systems biology approach. This is largely theoretical because it needs a major input in terms of funds, major input in terms of the kind of size, population sample that you need. And the drawback with this is that there's a great difficulty in getting the samples of healthy individuals. You have to compare it always. How will you get a signature if you don't have a comparison? So in order to get the comparative result, it is important to get healthy individuals. So there is and a lot of funding is needed. You need to do research at consortia level with big funding. But if this system is taken up in on a fast course way, it can give you genes, proteins, and metabolite signatures that are associated with specific nutrients and nutritional regimes, which can be catalogued, and they may serve as very good early warning molecular biomarkers. This is what is the need of the hour for nutrigenomics. You should have these early biomarkers so that they are prognostic markers, so that you can immediately say, no, this is not good for you, stop it. Or late biomarkers you can take in between turns to stop or change your diet pattern or nutritional regimes, or surrogate markers which can be indirect markers. So the whole idea of systems biology approach is to do population studies compared to healthy individuals and get an idea of those patterns which can be catalogued in order to come down to a very informative biomarker, which is warning biomarker, which is indicative of the onset of a disease. And molecular nutrition and nutrigenomics, they make a very good combination. You can discover dietary target genes and characterize the mechanism that that underlie the dietary sensing by the first approach, that is the traditional approach, in which you just use functional genomics to modulate gene expression. Or you can identify molecular markers that will allow early dietary interventions in order to reverse the effect of diet-related disease and bring back the regime, the normal cell homeostasis. Now, we've already seen that there are various levels at which the nutrients are going to impact the cell. And the levels of gene nutrient analysis for assessment of nutrient requirements can be at all these levels. The first two levels are absolutely context independent, whereas transcriptome, proteome, metabolome, physiome, phenome, and popular, they are context driven ones. And there are different kind of analysis which can be applied at all these levels in order to assess the nutrient requirement. So this is the first approach, the traditional hypothesis approach, and this is the major nutritional systems biology approach which is needed for the studies to be conducted in nutritional genomics. This can be initiated, and people have already initiated. This requires relatively small research groups and relatively small budgets, but this one requires the larger consortia and very big funding. So this is still in the pipeline, but here the initiations have already been made. So nutrigenomics, although new and not very well defined because very less convincing experimental data has been released, the expectations from this potential science are very high because it is directly related to human health. We need good data to support that our studies here. Now it's the right time that what we can do in the available budgets we should immediately start with, because unless we do start with the first kind of approach, we will not reach the second, which is a major one in which the markers are going to be markers will be designed. The application of genomics in nutritional science to prevent diet-related diseases is the main goal. So you know that if it is related to the human health, it has got the utmost significance in the priority charts. So nutritional genomics is a system approach to understanding the relationship between diet and health, and this will ensure that everyone benefits from the genomic revolution, and we should rather, because now there's no dearth of the kind of techniques and analysis tools which are available. They're all high throughput tools. Why don't we now put it to a better task to get a holistic view of what is happening between nutrition and diet and human health at the molecular interface. So we can get some better and more convincing interpretations of the same. Now, as I already told you, it requires a huge financial investments, and we have at international level, National Center for Minority Health and Health Disparities, Center for Excellence in Nutritional Genomics at UC Davies, United States, and German Berlin-Brandenburg Nutrigenomics Network, and in Netherlands there are 2 centers. And there's one KP6 program of the European Union, but India doesn't figure in anywhere in this yet. We have to make a start headway still. But at national level, we have started there are some private agencies which have started, which are a little closely related to nutrigenomics, although not exactly. They are gene support and they are nutrigenomics. They are joint venture between an African company, and it's based in Pune. They provide wide-ranging test panels to health-conscious individuals, and they're also able to predict the results or the risks associated with certain lifestyles. Right? So they do some testings and they predict this is good for you, this is not good for you. Then in Kerala, there's a Nutrigenomics which creates awareness of epigenetics, gene care, nutritional and gene therapy among the wider sections of society, those who have got awareness regarding this kind of, who are highly aware about nutrition and its impact on health. And they make scientific developments available to common man, those who are more aware and educated. And they ensure maximum quality of life for maximum period of time through personalized healthcare. The main idea is to give a personalized healthcare because every genome has got an imprint which can be identified through genome analysis as to how and in what way it should be treated. So main idea for all of them is to prevent rather than cure a treatment. You just capture it right from the beginning rather than start treating it later with medicines. So the definition of nutrigenomics, it started with study of effects of nutrients on the expression of individual genetic makeup, has now become more broad. Here now we can say nutritional factors that protect the genome from damage. We have already seen that they are damaging the genome of an individual. So what are the factors which we can include which will protect them? So this definition of the interaction between nutrient and the genes has now broadened, including the nutritional factors which will help us in protecting our genome from damage. So what can we learn from nutrigenomics? One important thing which we can learn is the dietary effect on the aging process. It's very important, and the study was a good indication of the fact that by calorie restriction we can slow down aging. Eat less, live more, that kind. So this has been done in the mice, and by doing so our normal lifespan of 75 can be increased to 105 with quality. You're not saying just living for extending our life, but with quality. Studies have indicated that as we age, the transcriptome has shown that the genes for structural proteins, for neurodegeneration, for carbohydrate metabolism, they show an upregulation, whereas fatty acid metabolism and mitochondrial function and gluconeogenesis, they get downregulated. And when age versus calorie restriction studies were conducted, they saw that age altered the expression of 996 of the transcript, whereas calorie restriction, it altered the expression of 21%, 11% more of the transcript, 2000. So what does it indicate? That the nutritional environment has a great impact on the gene expression than does the process of aging. So you can couple both of them and get the take-home message: eat less, live long. Eat healthy, of course. So calorie restriction can retard aging by inducing profound changes in transcription. And these changes in the gene expression, which are the dietary signatures, can be used to define specific effects of calorie restriction. Now they have seen that these are the dietary signatures which are produced as a result of restricting the calories. They were captured, and hence nutrigenomic approaches can provide markers that can be readily used to evaluate nutritional effects. So those signatures need to be trapped as per the kind of factor. Like here we are taking age into consideration, calorie restriction into consideration. So you can group them as dietary signatures for less calorie and increase of age, and you can use them as markers and evaluate the nutritional effects, right? Similarly, there's studies on the effects of selenium on certain genes. Here are the number of things which selenium impacts upon. And they have done studies with 4 groups of mice. They've given one group with selenium deficiency, then different kinds of salts in which sodium selenate or selenomethionine was given or Celplex were given, and the gene expression was compared in the intestinal tissue using MMS system. And what they observed was that Celplex, this altered the expression of 110 genes as compared to the other salts which were given. And the expression pattern was consistent with the low levels of oxidative stress in the tissue. So the kind of impact it did and the number of genes which were upregulated, both were seen with a specific kind of salt. So you can recommend this is better for this kind of oxidative, for combating the oxidative stress in the individuals. So now all this information is possible because we can analyze. We have the access to all these high throughput genomic tools. If we hadn't, we would just imagine the way Lavoisier was imagining. Yes, it could have a link with this. This could have an impact on this. So it was initially what was in their mind was just imagination. But these tools have now given clarity, or they have sort of identified with those imaginations in a more experimental and a result-driven way. So overall, they have seen the transcriptional changes induced by aging and selenium supplementation, and you can see what goes up and what goes down. So you can get a clear map or fingerprint of the impact of anything that you eat and do because of the use of the genomics tools. Similarly with long-chain PUFA or polyunsaturated fatty acids, which are involved in so many physiological processes. And here also alternate analytical platforms for identifying proteomes, networks or metabolomes, they can clarify the biological functions actually at a more detailed level with dietary signatures. So you can now measure the hidden nutrition, right? It was all hidden earlier. We didn't know what was actually happening in the body. Now you can see what is actually happening in the body at the genetic level, at the protein level, as well as at the metabolite level. So if you see the short-term goals of genomics, it is to identify the dietary signals, means the nutrition or nutrients that you take, elucidate the dietary sensor mechanisms, or elucidate the dietary patterns which will be produced, characterize target genes of these sensors, understand the interaction between them, and search for sensitizing genotypes, those who have got a predisposition to a particular disease. We will find the signatures in the form of gene expression, signatures in the form of protein expression, signatures in the form of metabolite profiles. So we need to know the dietary signals, we need to know the signatures they produce and how they produce and what are the new genes which are involved in this process. And what are the long-term goals? The short-term goals can all be achieved by traditional hypotheses, but the long-term goals are the ones which need a systems biology approach. This helps to understand how we can use nutrition to prevent many of the same diseases for which the pharmacists or pharmacogenomics are attempting to identify cure. We are at loggerheads with them. Instead of pharma, we are using Nutri here. With food, we are curing, not with medicine, right? That's the major challenge. And nutrigenomic researchers have the challenge to understand the polygenic diet trait. All these diseases like diabetes, like CVD, cardiovascular diseases, obesity, arthritis, they are all polygenic. So when so many genes, single gene involved, you can easily alter and you can make the difference. But these are all polygenic traits. So how does one move ahead with nutrigenomics with polygenic traits? That's the challenge. We cannot change our genetics, but if we know our weaknesses, we can create nutritional workarounds, right? We know my genes, I cannot change, but at least what I'm going to eat, with that I can create those workarounds by supplementing alternative pathways or withholding something from my diet so that my body can handle the disease disposition which I have. So the key objectives of nutrigenomics for the society are to diagnose and nutritionally prevent DNA damage on an individual by individual basis. So what is the need of the hour? Genome health clinics, like plant health clinics or various other human health clinics. We need genome health clinics now, a new mode of healthcare based on diagnosis and nutritional prevention of DNA damage and disease that result from it. So we need to have this, and you can have this. This is now upcoming in a very big way, because if you know your genome, you can make the you can work around your diet. And in the recent years, there have been a number of nutritional, metabolic, and diagnostic testing companies. For example, Genova, you have Metametrics. They have started selling genomic profile tests to help and guide decision making around dietary supplements. And all these tests are now becoming much cheaper. So we can all access, in coming times, we'll all be able to access these genomic profiling tests, and this will help in designing our own diets with more precision and carefulness, so that we can prevent the occurrence of a certain disease. Especially these typical diabetes, obesity, they are so prevalent and rampant in the coming times also, that we can take the help of all these, the Genome Health Clinic, to take all precautions. With the increasing lowering of prices, it's become very, very accessible now. Nutrigenomics, it aims to discern nuanced differences in the pre-disease states, such that personalized dietary interventions can be designed to prevent or modify future disease susceptibility. This is the main aim of nutrigenomics. You can, with all these tools, functional genomic tools for evaluating the effect of nutrition on the individuals, on the groups, on the populations, it will be possible to design diets that will meet the needs of specific individuals, as well as diets that address individual health problems that result from genetic determinants. So both nutrigenomics as well as nutrigenetics is taken into consideration. So the initial idea of having, there's so many diets which are prescribed. You take this, it will help you in this way, but not every diet impacts each and every person in the same way. There's no one size fits all. Now is coming the time or the age of individualized nutrition, your personalized nutrition. You can eat right for your own genotype. If you have the card of your own genome with you, your doctor can now devise the diet which is going to suit your genome, right? And that is going to be the future. It's a very ambitious challenge for the next decade. And here the translation of nutrigenomics data into an accurate prediction of the beneficial or adversary health effects of dietary components will be analyzed, and based on this, provide a diet, a lifestyle, and drug recommendations to individuals to maintain their health and to prevent the diseases, rather than simply develop diagnostics and drugs to identify and attempt a cure to them. So the idea is to prevent rather than cure, and prevent with food. So another visionary Hippocrates, who had said, leave your drugs in the chemist's pot if you can heal the patient with the food. The first aim is to treat the patient with the quality food, good nutrition, and the nutrition which suits that person, rather than attempting to go in for the drugs. So your genome gives the DNA information to prescribe that personalized diet. So this is how we are going to link the nutrition with the genome in the future and design our own plates, whether it is breakfast, lunch, or dinner, to keep ourselves more healthy and live longer. So all these kind of insights are now possible. Time is absolutely ripe to make interventions, very, very vigorous interventions into this area. because it has got very high potential as it's the human health which is concerned. So if our policy makers, they take a good stand or they recognize its potential, then a lot of funding should go in this direction to promote this kind of science, this nutritional science branch. Thank you so much.
So now the talk is open for the discussion. If you have any queries or questions, you can come in front and ask.
Thank you for your informative talk, ma'am. I have a couple of questions. First one is, as my knowledge on genetics, I have studied there is a link between telomere with aging. As age progresses, the telomere length will be decreased. So there is a correlation between telomere and aging. In one of your slides, you have explained about the correlation between calorie and aging.
Yeah.
Is there any studies on relationship between this calorie on the telomere?
Not yet.
Okay.
Not yet. There is none. This is a very general fact that the less calories you take, the less you irritate your metabolism and you can live longer. And the kind of food that you take, that also impacts the kind of lifestyle you have. The kind of lifestyle you have, that is going to impact your kind of your aging process as well. So as far as telomeres are concerned, it hasn't. But this is a fact. You are right about it. But the relationship between telomeres and calories has not been.
Okay. And second one is application point of view. As per this Nutrigenomics, the most application point of view is personalized diet or personalized nutrition. During past decade, people were talking about they have shifted from generalized medication to personalized medication. This is more successful in developed countries. And what's your perspective of using this personalized nutrition in case of developing countries?
Yes, we should in fact, because in our developing countries only, all these the incidence of diabetes, incidence of obesity, it is on and rampant. So we need to take very active measures to control it at this stage. But we don't have the policies to do those consortia system biology approach, so that we can really go on a fast track for developing certain markers that we can control the obesity. What do you say, which we are predicting, the obesity number which we are predicting in 2050, 98% of diabetes we are predicting. So we are actually, the developing countries should take very, very quick measures and put in all the fundings for starting this kind of research at a priority level. We should, in fact. We haven't still started doing research at that level. So it is the need of the hour for the developing countries rather than developed countries.
As per I understood, we need a whole genome to have this nutrigenomic study, ma'am.
Yeah.
Okay. Actually, I'm very curious on sequencing my whole genome, but since even we are in a research field, I don't know where to give my sample to get my genome sequenced. Then how about the illiterate people or the people in
So
rural
that's
area?
what I'm saying. We should be on a fast track. The government should be on a fast track to make this kind of science popular, because it has a lot of potential. We should all have our genome sequence in our pockets. So make it cheaper so that people can do that. In US now it has come down to very $1,000 or so per person, or maybe lesser now. So we should all look forward to that kind of style, that there should, and it's not that we can't do it. We have very potential people here, potential laboratories here. So it's just that the priority should be given for this kind of research, and we will be on the right track after that.
Thank you, ma'am.
Yeah.
Anybody else? Yeah, please.
Thank you, ma'am, for the talk.
Thank you.
I would like to ask, what could be the strategies which would address the development of personalized nutri-therapy for the diseases which are controlled by multiple genes, like hypertension, or these diseases are majorly controlled by multiple genes. So, what could be the strategies to address the development of personalized medicines for those, or if there are any studies going on in India also for that?
Not yet, not in India, but for the strategy which you are asking for polygenic diet diseases, they should mainly involve the systems biology approach, in which you should take the populations into consideration and develop the markers, early markers or late markers or surrogate markers, so that you can predict, or which can give you a warning signal that yes, this is not good for this population in this area or for this individual. So for such kind of polygenic, it's best to have a systems biology approach, so that we can develop good or markers, accurate markers for indicating or warning the individual suffering from them. Not the first.
Is this is operational outside India?
There are those consortia, I said,
They
who
are
are
actually including
yes,
multiple gene diseases also.
yes. But the markers, I don't think, are still out, because the work has initiated, but the markers are not out yet. And moreover, they will be studying their populations. Our populations, would be different. Environment is different, so we could have a different impact. We are not even a part of their consortia yet, so but we need to do it. Yeah,
Thank you.
thank you.
So, please come. So most probably this will be the last question, and then later on you can discuss.
Thank you, ma'am.
Thank you.
My question is related to biomarker. Ma'am, any reliable biomarker related with the nutrigenomics, and what is hurdle between the developing the biomarker with the in nutrigenomics?
I told you it needs to analyze a very vast population,
Yes.
right? But for reference, you need healthy individuals as well. When you develop markers, you need a reference also. The problem is developing or roping in healthy individuals. So these glycosylated hemoglobin is one of the marker tests. Then people also do micronucleus assays for genotoxic effect of certain carcinogens on human diet. These are the tests, right?
Yes,
and the markers are for surrogate markers, which are indirect markers, like I just said, acetylated hemoglobin, which will 3 months this thing sugar you will do, and you will get an indication that it has been on a high, right? So you can stop, or you can say the person is prone to diabetes. So these kind of markers are available, but this is just one marker. We need to develop many more markers like this, and individuals will not be enough. Populations are needed, yeah, and populations under different environmental conditions are needed, so that you can give, yes, this is one marker, it will stand true for all the people around, or this group of population. That really needs a lot of conviction and testing before it is released as a marker.
Okay, thank you, ma'am.
So if we can have a DNA barcode for the identification of genotypes, so definitely this science is picking up, and very soon, so all the myth which has been forecasted, that will become reality. So I think Madam has nicely explained almost all the dimension of this upcoming science, that is nutrigenomics. So since it's picking up, so many unanswered questions are there, but then definitely, so we'll find the answers in days to come. So with this brief note,
In decades to come.
in decades to come. So with this brief note, now I request my coordinator to please felicitate Dr. Archana Sachdev ma'am for giving such an interactive and interesting talk.
Thank you so much. Nice. Yeah.
And thank you, ma'am, for
Thank
giving
you.
such an interesting.
Thanks for your patient hearing. Thanks.